Sanbonani, welcome. My name is Gosisitole, and I am from the township of Impopomeni, which is outside the town of Howick in the Midlands of KwaZulu-Natal. This place is known for the mighty Howick Falls and the Nelson Mandela Capture Site. It is a place of green rolling grasslands, farming communities, creative arts and crafts, and natural beauty. Our special township was once a mosaic of beautiful wetlands and grasses. The water that runs through the streams here go directly into Midmo Dam, which stores water for a large portion of KwaZulu-Natal. Unfortunately, you can see that often we have issues of overflowing manholes and water pollution. As a group of EnviroChamps, we decided to investigate what has changed in our environment and has led to these water issues. We chatted to the oldest members of our community and learned that this place has not always faced these issues. We also realized that people in Africa have always practiced citizen science, even though it was not called that until recently. The Nguni people in southern Africa, for example, collected water from a stream where they could hear the water. Water that is moving and bubbling over rocks and stones in the stream is more likely to be healthy than stagnant water in a pool. When collecting the water, they would sweep the surface water with their forearm or with the collection parts before lifting the water. Amanzi mnandi, sweet water, out of a hole they had cleared in the surface water. This process removes surface water and dust that may be on the surface of the water. The isiXhosa people cleared spring water using an ancestral practice of sprinkling wood ash on the surface, particularly during the rainy season when the water became cloudy. Wood ash is a flocculant that enables small particles of silt to be drawn together and sink to the bottom of a pool. Wood ash flocculation would clear a pool so that it was possible to collect clear water, as well as to see the bottom of a pool for evidence of healthy organisms living in a village spring. Did you know creatures that lived before the dinosaurs can tell us how clean our rivers and streams are? It is surprising that wherever humans live on Earth, their local streams have many similar organisms. These include dragonflies, mayflies, beetles, and snails. Scientists call these small creatures, which are visible to the naked eye, aquatic macroinvertebrates. Scientists have discovered that these nunus lived in rivers and streams before the time of dinosaurs, when there was one megacontinent on Earth. This was long before life as we know it today was present on Earth. The first megacontinent was known as Pangaea, and after many millions of years it split up and the continents drifted apart. These formed other megacontinents like Gondwanaland, which eventually became the continent of Africa. Interestingly, the small locally adapted water organisms traveled on the continents and became scattered all over the world. This means whether we live in the warm tropics or the colder temperate regions of the world, we find these animals in our freshwater streams and rivers. They also occur in the streams in the low-lying areas as well as high up in the mountains. This is an amazing fact. What this means is that any class in any school anywhere in the world can read and assess water quality using the organisms that live in their local stream. Scientists have discovered that these aquatic macroinvertebrates react differently to pollution, and they have given a sensitivity score to each group of nunus. People have adapted this system to develop the mini stream assessment scoring system known as miniSASS. So how does miniSASS work? First, you need to collect a sample of freshwater macroinvertebrates from a stream. You can use a net or any containers you have. Then you use the key to identify which groups you have in your sample. There are only 13 groups to choose from, so it's really not that difficult. Next, you calculate your average score, which will tell you which category your river falls into and if it's in a good condition or a poor one. As you can see, in this miniSASS assessment, we identified worms, crabs or shrimps, minnow mayflies, dragonflies, bugs or beetles, true flies, and snails. From the sensitivity score, we calculated our total score to be 30, and the number of groups of the macroinvertebrates that were found were 7. We then calculated our miniSASS score by dividing our total score with the number of groups. As mentioned earlier, we moved into our ecological category where we identified the state or the health of our river. Our river was rocky, and because our miniSASS score was 4.2, less than 5.3, this tells us that our river is in very poor condition. Thereafter, you can load your results onto the miniSASS website, which is demonstrated in this video. People from all over the world are now using miniSASS, as the groups of invertebrates are found everywhere. We even use this method to assess the quality of the Orange River, all the way from the source to sea. This project involved 4 countries: South Africa, Botswana, Namibia, and Lesotho. miniSASS can help us monitor what is happening to our rivers. We can see where pollution is being added just by looking at the map on the website. Here you can see how the color of the crab symbols change after the place point source pollution enters the Umgeni River. Purple and red show the river is in trouble, while green shows that the river is in a good condition. We can even use miniSASS to track how we are working towards achieving our Sustainable Development Goals. It is a direct measure of SDG 6.3, which is focused on water quality. After discovering all of this, our team of EnviroChamps have decided that we are going to use miniSASS to monitor the changes in the streams in Mpophomeni. This will help our municipality to improve the quality of water before it goes into Midmar Dam. Already, this work has initiated the building of a new water treatment works in our township and the wetland rehabilitation adjacent to the dam. It's amazing what we can do or what can be achieved through participating in citizen science. We look forward to seeing what you do in your community, so share it with us. Thank you for your time.